Okay, so let me formally welcome very heartily our distinguished guest. It is a great privilege to have you here and a great opportunity for all of us to ask you some questions about the career, about specific aspects of your work. So I hope that it will be very fruitful and quite pleasant for you as well. And let me also welcome all our guests, our rector, vice rectors, and academic staff and students. I think what I was able to see from top to the bottom, so both the advanced researchers and PhD students as well. So I think with that we can formally start our meeting.
Okay, thank you. I'm wondering if we could turn on the lights, because you don't know, but you're all dark. Can't see anybody there. So is there someone who could turn the room light on?
Just a second.
Proszę zapalić światło na widownię.
Yeah.
Okay.
Okay, great.
Thank
Shall
you.
we sit down?
Yes.
Yeah.
All right, thank you.
So maybe just to set up a scene and make other people comfortable, I would like to start with the first question, maybe something very usual in such situations. Can you identify some turning points that made you choose the specific path of career and then to undertake the specific area of research?
Well, you know, when I began my undergraduate studies, which was in 1971, you know, I went to university and I wanted to be an astronomer, but I met some other students in the dormitory where I was that were second-year students, and they had been working in laboratories, and they were so excited about the work they were doing in molecular biology and genetics that I was infected by this, you know, enthusiasm for biology. And so it was really other students who turned me to what has become my passion all my career. And at the time, people were using genetic approaches to understand primarily bacterial cells and using, you know, bacteriophage genetics. So the genetic approach to biological problems was one that I got, you know, just so excited about. And so that affected, you know, sort of my orientation thereafter, to try to seek laboratories where people were doing genetic, certainly molecular approaches to biological problems.
Okay, and what about this specific focus on microRNAs?
Oh, so my focus on microRNAs was not a result of any kind of rational choice of my own, but I went to Bob Horvitz's lab to do postdoctoral research. But my aim was not to go, I did not go to that lab to work on any particular problem. I went to that lab because Bob Horvitz was someone who inspired me, and I was ready to learn from him, and I was open to whatever suggestions he might have for projects. And so he suggested a study of these mutants that had developmental timing defects, you know, as they developed. These were worm mutants, Caenorhabditis elegans mutants. And so it was basically on, you know, my postdoctoral advisor, Bob Horvitz's advice, pointing to me to these mutants. And the mutants ended up leading us to microRNAs.
So in that there's some hope for us who don't really have the specific focus on anything, that something maybe will pop up in the future.
Yeah, I try to tell these stories as a serious example of how, and it's not uncommon, right, that we are led where we go scientifically by initial choices that lead us in directions or to other problems that we didn't anticipate. So we may be answering a different set of questions after we've embarked on a project than what we started the project with. Yeah.
Of course, any questions from the audience are welcome.
So as you can see, that there is not as far from the astronomy to the molecular biology. Actually, we can say this is really quite near from these different fields of research and science. And maybe we should, at the very beginning of the scientific career, choose some of the icons, we can say, and maybe we should go after them, and then maybe we can be flame out or start on with any other aspects. So we should focus, but be also open, very open for any ideas.
Right. I think that's the important concept, is being open, because especially in biology, there's so much that we do not understand, right? So that when we try to approach a problem, the uncertainties are often not even clear yet. So we need to be open to our project going in some direction that we didn't anticipate, but also open to colleagues giving us guidance. And now it need not be an icon. I think Bob Horvitz would say that at the time that I joined his lab, he was not an icon. He was just starting his lab. He was a junior faculty, you know. So, but that's not to say that if you have access to an icon, then you should, you know, try to draw on that person's wisdom. But I'm here to say that much of the guidance I've received over the years has been from peers, you know, or for example, other students, other faculty. And then, you know, suggestions when you go to a meeting, you know, a symposium somewhere, you learn. And then also from visiting, like, for example, here, you learn about fields that are unfamiliar, and then that's really stimulating, and that can guide projects as well. So openness, is a very important principle. Well said.
Yeah, today we saw during this scientific meeting that the biotechnology is also multiple interdisciplinary and combine all these different fields of science, and we can meet all together in one specific point.
Yeah. Yeah. Somebody must have questions. Are there any brave graduate students or faculty?
Okay. Hello, good morning, good evening. So first of all, I would like to ask you two questions, because you started from telling us how much you have been inspired by the Bob at the very beginning. So if you just could tell us something more about his personality. And second question is, how far or how close is from astrology into microbiology?
Oh, from astronomy to microbiology? Yes. Yeah, right. Molecular biology, right. Yeah, okay, the first question is about Bob Horvitz. He was clearly dedicated and believed deeply in the principle that we can learn new biology, you know, and completely new principles from studying, a very simple animal, this Coenobdinus elegans. And he also believed, moreover, that that animal would teach us about human biology, and which was, I think, quite a visionary sort of perspective to have at those days, because there was no genome sequences that nowadays allow us to just see molecular homology across genomes, right? So at that time, they had worked out the complete cell lineage of C. elegans, meaning they could describe the development of this animal from one zygote, single cell zygote, all the way to 559 cell larva, and then that larva would develop through 4 larval stages to about 1,000 cell adult. So when it's at its most complex, it only has 1,000 cells, and they had actually worked out, he and John Sulston had worked out that lineage. And so that was a resource that allowed people to study developmental problems on the level of the behavior of individual cells and individual cell divisions, because you could watch them, all those developmental events occurring in the animal. So Bob, sort of conveyed to us this conviction that we were going to learn new biology from studying this little animal. I think that was probably the main thing that captured my imagination. And he was also, you know, a very good geneticist. And we felt it was straightforward to learn from Bob the elements and principles of genetic approaches, because he was so, he had such clarity, let's say, in how he expressed how to approach experiments. And the second question was about, and Bob, by the way, is a very generous person too. So he allowed me and Gary Rufkin to take our projects with us, you know, and we were his first postdocs. So that was really, you know, very important. And he actually Bob let me publish a single author paper about work that I had started in his lab, but, you know, most PIs would have just automatically put their name on that paper. Yeah, astronomy and molecular biology. Well, you know, I think later in my career, I've come to appreciate that both astrophysics, astronomy, and cosmology and life sciences have been, over the past, you know, 3 or 4 decades, sciences that have been changing so rapidly, right? Theories are even being challenged, you know, in astrophysics, you know, dark matter, the acceleration of the expansion of the universe, you know, dark energy, you know, these kinds of phenomena that we can't explain, you know, and then now with the Webb telescope looking farther back, and we have to challenge presumptions about the evolution of the early universe. So likewise in biology, I think we've seen, you know, repeatedly over the decades, new phenomena emerge, you know, RNAi, you know, and technological advances as well in parallel with these, you know, with the studies of the universe, studies of living things. So we can go now deeper and deeper and deeper. For example, now I'd say the analog of the Webb telescope, where we can look farther than we could before, cryo-EM is a way sort of that we can now look at structures at a deeper detail, sort of more comprehensively, you know, than before. So it's been really enjoyable sort of to be as part of science broadly over the past, you know, 3 or 4 or 5 decades. But I still love astronomy and astrophysics, and it's really been fun to observe that both sciences have been extremely active in turning up and turning over, you know, new ideas.
Hello, I would like to ask you about microRNAs, because recently, like in last years, the number of microRNAs is increasing, and now it's like thousands of microRNAs. What do you think? Do you think that they are really all microRNAs, or are there many false positives? Because in base, it is like expanding really quickly.
Yeah, this is a really interesting question. You know, most recently, in my group, we've come up against this question in the process of documenting and annotating the microRNAs in a new experimental organism, or an organism that we're taking up, which is the acele worm. It's called, or maybe the striped panther worm. You may know it's not a flatworm, not a planaria, but it's actually very distantly related to the planaria. It's actually a basal bilaterian, right? And it can do whole body regeneration. So the postdoc who's working on this wants to study microRNAs in regeneration of the whole body, right? And so he's been doing cloning and sequencing and documenting of microRNAs, and he came up with, I think, at least you know, more than 1,000 microRNA genes in this organism, which is many more than in other organisms that have been analyzed. But part of the reason is because his data sets are very, very, very deep. So he's done, you know, hundreds and hundreds of millions of reads from multiple developmental stages. So what we're confronted with is the reality that there's a small RNA maybe produced from the genome, right, from a hairpin structure, but many of those are only found in this animal. So we wonder, could they be functional? Are they just genomic noise, right? Are they incipient microRNAs? I'm sure you've thought about this as well, right? Could this be a microRNA that's being expressed in it's under a certain amount of selection in the organism at the moment, but remains to be seen if it's going to be fixed in evolution? Yeah, they're really interesting questions. And so we had to decide in writing the paper where to make cutoffs, right? And then below that, we've got more, and he's saying, well, I'm not going to call them microRNAs. I don't know what to call them yet, right? But they may be a class of small RNA that is functioning in ways like microRNAs or different from microRNAs. We don't know. I guess one of the things we can do in what organism that we're working on is to pull down the argonaut by immunoprecipitation and perhaps clone what's associated with different classes of argonauts, and that could be a way of organizing them. So for example, in Cedarmoditis elegans, there's 27 argonauts, and many of those argonauts are involved in endogenous, gene regulation, the silencing of transposons, genome defense, and so forth. And some of those small RNAs are, restricted. They're restricted to certain argonauts that are devoted to these functions. So that could be helpful as well. I love talking about microRNAs. I could talk and talk and talk about them.
So maybe let me ask a more philosophical question. You have mentioned that despite the rapid progress in molecular biology, there is still a lot of things that we don't know. So do you believe that there will be one day on which we will learn everything, or will we be always on the path of expanding knowledge, but never getting at this point of knowing everything?
Right, that's a very good question. Yeah, I mean, I love philosophy, and my view is that that it'll be a very, very long time until we actually can claim to, let's say, know, most of what we need to know about biological systems, right? I mean, because the problem can keep expanding. I mean, I feel like it's important to try to understand as quickly as we can human biology, right? And so perhaps there'll be a time when we'll have a reasonably good description of the human biology. It's hard to imagine that we'll ever understand, consciousness and cognition, but perhaps we'll, know the rudiments of that. This is something that reminds me, I'm reminded of two things, two points. So one is that I think that we are programmed by evolution to assume that we know everything or most of what we need to know to function in whatever context we happen to be contemplating, right? So in the context of this room, I assume I know everything I need to know. I know that you are all friendly, so you're not a, not a threat. I'm assuming I know the building is well made, and so the roof won't fall down. I know about the atmosphere, so the room, the air's not going to be sucked out. So all these things we assume about, the chair's going to hold me because it always has. So I feel like we're conditioned to assess our environment and kind of know, okay, I know what I need to know in this context. And so now when we study something we can't see, right, I think we are always, whatever we learn, I think our instincts tell us, oh, so now we kind of understand this thing as, pretty well. And the concept, the idea that, let's say, most of what we would like to know, most of it is unknown, right? That's a very unsettling idea in the real world, but in the scientific world, that's the most exciting idea. And I think but we have to actively remind ourselves because it's against my instincts. So I keep reminding ourselves, 500 years from now, we might have a meeting just like this and people are sitting around thinking and asking that same question, So the other thing that reminds me is I have colleagues at UMass who they work in therapeutics development. And so this would resonate with many of you, you know. So mostly it's RNA therapeutics, you know, RNAi or oligonucleotide therapeutics, and they try to figure out ways to deliver these reagents to particular organs or particular cell types. And they work on trying to do this, and I often are sitting on the committee of a student, and the student is trying to, you know, for example, cure glioblastoma, by like targeting, let's say, a histone variant that's underlying many of these things. And it seems like on paper it should be straightforward, but everything she's trying, you know, is not working, right? Or something works and then something doesn't work, and it should work based on what we think we know about the system, you see? And this is happening all the time, almost in every project, therapeutics project, you know, in these labs. And what's happening is we're trying to engineer a biological system that we have probably very poor understanding of, right? But those therapeutic development efforts actually allow the investigators to identify, you know, areas that we need to know more about, right? So, you know, why doesn't this reagent go into the tumor cell, but it goes into the normal cell, right? Or because, you know, and why does it go into some of the cells in the tumor, but not the ones you want to, you know? And well, we thought it was engineered to go there, you know? So now they're trying to figure out, you know, why, and that's going to reveal more about fundamentals of tumor biology, right? The act of trying to engineer the system forces us to learn more about the system. So I think I'm reminded of that as we sort of are discussing the limits of our knowledge, you know, at any given moment.
Okay, I think there should be some.
Oh, another microRNA question.
Hello.
Yeah.
My name is Aleksandra Maksimowska, and I have a question from some other perspective, because apart from being a student, I also help students to learn and practice before their final exams. So what would be one piece of advice that you would give students before their final exam this year? And also, what is one thing that you would say to those who would like to pursue such a path as yours, or such a career as yours? Scientific.
Okay. Yeah. Advice when you're preparing for exams, I would say do not do it the way I did it. I think I failed to study, I failed to utilize my colleagues, other students, right, when I was studying. In those days, for some reason, I believed you had to study alone, you know, but nowadays, when I visit a university, I see students working together at the same table or something. So I would say, yeah, working with other students, studying together, really sharing, you know, knowledge and trying to explain things to each other probably is a really great way to help reinforce your own understanding. When you try to explain it to somebody, I think always we become better, you know, we understand things better as we make that effort. And if we can't explain it, then that probably means we're missing something. And then doing that in a group, I think, would be super, super helpful. The other thing is try not to sleep through the beginning of the exam. Because at least once I remember showing up for the exam 2 hours late for a 3-hour exam, right? And I thought, oh, it's not at 11? Oh, you know, so the other question was advice about people going, let's say, on the path like mine.
Yes.
So we'd have to define my path, let's say. I mean, but I would define my path as a basic science focused career, you know, where I've never really developed any expertise in approaching a problem starting with, say, a disease or with a physiological context. I've mostly used genetic approaches, using a model organism. So I would encourage people to think about going along a career where you're going to study basic problems of biology, whatever they may be, you know, using some model organism, some genetic approaches. Now, in today, you don't necessarily have to work with a model organism like C. elegans or Drosophila or zebrafish. You can actually do human genetics, as you know. So nowadays, there's a whole other fascinating and important dimension to genetics where the human genetics, you know, genetic diseases, rare diseases, you know, genetic contributions from, you know, GWAS studies and so forth. You can begin with the gene, right, in the human context as a lead. And in our lab recently, we did some projects where we were working with colleagues who are human geneticists, and then we used C. elegans as a model to examine the consequences of some of those mutations. So the human families presented the mutations, and these are mutations in the microRNA Argonaut. And then we tried to contribute to the project by taking the exact same mutations and introducing them into the worm Argonaut because the amino acids are conserved, you know, and that was very interesting. So, yeah, that's a long way of saying model organism genetics is alive and well today as ever, but also there's a wonderful sort of partnership, let's say, between human genetics and model organism genetics that is really exciting.
Thank you so much for the answer. And of course, if that's okay, I would share it with my students on social media, perhaps.
Yeah.
Thank you so much.
Okay, thank you.
Okay, I would like ask also the, let's say, philosophical question. What do you think? Cells are altruistic or selfish? And in what sense they are altruistic? Because I am thinking about apoptosis, for example. Is it altruistic or not?
So are cells altruistic or selfish? Yeah. Yeah. Well, it looks like it depends on the context, doesn't it? I mean, it seems like a tumor cell. It strikes me as selfish in the extreme Darwinian case, right? But of course, tumors are an environment where there's an ecology going on there, right? So, yeah, it's a very so. It's a really interesting question. It's not only a philosophical question, is it, right? Because it has to do in a way with, let's say, what is the potential of cells, right? What are the biological potential? What's the potential in terms of functionality as a particular cell or as a population of cells, a tissue of diverse cells, you know? So I think the more we learn about biology, isn't it the more we learn about the vast potential, right? We start to learn about, what was I learning about the other day? Oh, I was talking to neuroscientists and just learning about the amazing complexity of you know, circuits and functionality in the brain that they're beginning to uncover now with new technology. And they were conveying to me the amazing surprises they're finding. So they constantly find surprises that shake up sort of assumptions. And almost always those assumptions are, what's shaking them up is revealing more potential for complexity than they had anticipated. Yeah, so that's and that's not unrelated to this question of where we'll be in 500 years, right? You know, because the complexity keeps unfolding as more and more complex, and so therefore more and more potential for. And if we just look at all the animals out there, an incredible diversity of animals, my God, and all the animals that have ever lived, you know, and they're fossilized and so forth, the diversity is so incredibly astonishing. And underlying all that has to be perhaps almost a limitless resources of ways that cells can compute as individual cells and collections of cells and so forth. Yeah.
So I have another microRNA question. I would like to ask about microRNAs as therapeutic targets. There are a couple in clinical trials, but do you think they will be used really in therapy? And do you think that it will be in 10 years or less or more? And which of the candidates is the most promising according to you?
Oh, so what did I hear about recently? Regulus was acquired by somebody, and I think Regulus has a miR-34, I think, target. So they want to target miR-34 in the context of, I forget what it was. But it was good to see that there was some activity in this area, meaning a microRNA targeting company had got acquired by a big company specifically to acquire those programs. I mean, as you know, it's been kind of slow to let's say enable some sort of therapy where one would either be resupplying a microRNA that's missing in a you know, pathological or physiological context, or to inhibit a microRNA that is hyperactive, right, in that context. Maybe some of the complexity is related to what we heard about this morning, where, like, say, miR-155 can be a tumor suppressor in some context and an oncogene in another context. And so the complexity of maybe the pathways and networks that these microRNAs are embedded in is often maybe is what people have come up against, and that's related to this principle of, as we try to perturb systems, you know, what we don't know about the system reveals itself. So perhaps that's part of the reason why these therapies are you know, slow to come to realization. But it's promising. You know, of course, the RNAi therapies, which is not unrelated to microRNAs, it's the same Argonauts and you know, the same small RNAs. Those have been more effective and more fruitful, and there's more of them actually in approved, because it's so, the specificity is so high, because you're using that microRNA or that small RNA to kill a certain mRNA. The microRNAs have this looser connectivity and therefore more targets and more complexity, and perhaps more confounding issues biologically as you try to target them.
It's me again. So my question is, because I believe it was 1993 when you published your paper with your discovery, and if you would divide your scientific path into two stages. The first one before 1993. So what was the motivation to take this run and to find out about your discovery, and how many times did you fall? And second one after 1993, when you have to wait more than 20 years to become the Nobel Prize winner. When did you start to believe that your discovery is so huge and so fundamental?
Well, first of all, I never believed that this discovery was, Nobel Prize worthy. I mean, that's despite the fact that me and Gary Ruvkin got lots of other awards. But we felt that it was probably that we were lucky to get these other awards, but the Nobel Award that's another sort of, order of magnitude more impact, because, so many of our scientific heroes were Nobel Prize winners. David Baltimore, Richard Feynman, even Bob Horowitz, and John Sulston. These were people who worked on worms, but we looked up to them as extraordinary scientists, right? Now, in 1993, we published this paper. We didn't really, at that point, feel that necessarily this was something. We found a microRNA, but we didn't expect that it would be necessarily an example of something that was found even outside nematodes. And, Gary Ruvkin then later found LET7 in 1999. So it was 6 years before even the second microRNA was found. And that was, again, in C. elegans, you know. But you asked me, like, where my career might be divided, you know, between before and after. It was when Gary Ruvkin found that LET7 was conserved in all the bilaterian animals. So that means LET7, that microRNA, evolved like 600 million years ago, right? And it's for some reason, all the animals since then have had LET7. So that moment, actually, when I learned about that finding was oh my God, so there must be more microRNAs and they can't just be in C. elegans. Yeah, so that, and after that, you know, we found some more, then Bartel and Tuschl, and then plenty of other labs were working on it. And then after that, when I would go to meetings and all my scientific heroes would be there working on microRNAs. My PhD advisor, David Baltimore, is working on microRNAs, you know, and folks whose biology, you know, and research I admired for a long time were now studying microRNAs. And I'd sit in the audience and say, you know, wow, this is unreal. You know, it's really, really crazy. But then, you know, of course, RNAi was discovered by, you know, Craig Mello and Andy Fire, and they got Nobel Prize in 2006 for that. And Gary and I understood, of course, we're not going to be part of that because there's too many of us. There's me and Gary and those two, and what importantly, what Andy Fire and Craig Mello had discovered, they had discovered that microRNAs were part of RNAi because they used the same argonaute, right? And they were produced by the same dicer enzyme. So that Nobel Prize for Fire and Mello was really encompassing these phenomena that involved microRNA and RNAi. So we considered that a totally appropriate, you know, prize. You know, those guys made that discovery. So, you know, people would come up to us. I'm sure it happened to Gary. People would come up and say, "Oh, you know, someday you're going to win the Nobel Prize." But I never believed it. Yeah, and in fact, it was something that I dreaded. Like, it would be like a bad dream if it happened, right? Yeah, because why? Because I'd feel like it was a lie, like I'd be an imposter. Because, you know, it's just like, you don't think of yourself in that context, right? And so when it happened, it was just like now again changing my life, you know, in a moment, because I had to quickly reconcile in my own brain, all right, this is okay. You know, you're not an imposter, right? This is not a nightmare. It's not a lie, right? And so, and the reason why I was able to accept it is because these people on the Nobel Committee they're not fools, you know. I mean, you know, they know what they're doing. They read all my papers. And I met the lady who was in charge of my file when we went in December, and she's more of an expert on my work than I am. It's unbelievable, you know. And so you look at her and you say, all right, they must be right, you know, and this is great. But what I never forget, right, is that it was an accidental discovery, right? And what I love about science is there's many approaches one can have and be successful, even as successful as I have, by not being particularly brilliant, not being visionary. I just happened to be in the right place at the right time. I was Bob Horvitz's first postdoc. I'm the one he happened to give the project to, right? And I didn't identify the project and say, you know, Bob, I want that project because there's a lot of, you know, interesting stuff there and it's going to be awesome, you know. It's like, oh, but Bob says this is an interesting thing to do. I said, yeah, it looks good. Okay, yeah, I'll do that, right? Well, you know. So you see what I mean? Like, I'm just like a regular person who's, you know, lucky enough to be in the right circumstances, and you know, we made this discovery. But my goal was never to do something like this. It was really just to be in science and be a scientist, and you know, and be with scientists, you know, and try to make a contribution. So when people ask me, like, you know, what would your advice be to a young scientist? So how would you proceed so that you can eventually win the Nobel Prize? You know, I mean, I can tell you, I am a counterexample about how to do that because it shouldn't have happened to me. So, but the important thing is to set your sights on just being a good scientist and making a contribution, right? Because that you can achieve, right? But you have to make sure that that's what you're going to get satisfaction from. I've completely forgotten your question. Is that even close?
But you answer much more questions than I expected to. So, but it cannot be such easy.
Yeah,
it cannot be.
No, it's not.
So you have to start to believe in yourself.
No,
maybe it is the moment.
Yeah, no, I'm here to say that. I mean, maybe one of the important lessons that you can take from a story such as mine is that, well, yeah, I had the opportunity to work in these labs of undergraduate lab, then Baltimore, and Horvitz, and then I I got good jobs in good places and stuff. So, but the point is, I had the opportunity, right? And so what we have to remind ourselves is what's in short supply in the world is opportunity. What is in plentiful supply is talent. So there's far more talent than there is our opportunity. And so that's what I'm reminded of when I sort of, contemplate my own career.
Thank you very much.
I have one question. Can you tell us something about your research group? Who is there, and how do you work with the people? How do you motivate them, and how the work is organized? How do you manage this?
Yeah, I've always had a fairly small research group, usually a mixture of half postdocs, half graduate students. A common thread in my career has been certain graduate students have been particularly important to energize my research at different junctures. Like the first graduate student who launched what I was doing in the lab, as well as certain key postdoctoral fellows. At the same time, I've always been as through my career I've become increasingly open to different options, career options that let's say students and postdocs can do. Initially, when I first started out, I was assuming that if my student or my postdoc did not go into academia, that would reflect a failure on my part, right? So now that was, poor thinking, because of course it's up to the student to choose their path, or the postdoc to choose their path based on their inclinations. But also, as all of us know, opportunities have expanded for people doing a PhD or doing postdoctoral work. They can go into other than, there's biotech, there's law, there's education. And so by the time the first student who came to me and she said, I want to be a high school teacher, right? So a graduate student. By the time she came to me, I welcomed that, right? And she had taken almost a year and a half to get up her courage to tell me about this ambition of hers, because she thought that I was going to assume that that was a failure on her part because she wasn't going to be. So that was an important step in my career, because I was able to tell her, no, my God, I think this is awesome that you have this vision that you're working towards, And she went on to be an extraordinary high school teacher. So, I think that, so I've tried to become more aware and more, open to people choosing their own path. I think, the actual research direction of the lab is for the most part has been governed by the interests of the people in the lab, right? So I've had an NIH grant, National Institutes of Health grant, for 40 years to study C. elegans, this little animal. And the grant changes each cycle a lot, which is based on what people have been doing in the previous cycle, which is often not very closely related to the proposal for that previous cycle, if you know what I mean, right? So you have aims, plans, and then people are joining the lab and doing things, and projects are evolving in the way that we discussed earlier. So by the time the grant is being renewed, the aims will have all changed, and sometimes the direction has changed, you know. And that was based on, you know, people's interests and the way their project evolved in the lab. So I'm not very good at determining, you know, programmatic directions and themes, you know, and implementing that. And it's more, we follow the way things go based on the way each project has moved and how people's interests have evolved.
Hello. First of all, congratulations on getting this prestigious award as a Nobel Prize. I have one simple question. How much this Nobel Prize changed your career? It's been a year, maybe less, but do you see an impact, a change in how do you conduct research, publications, or is it just a prize, a huge achievement, but it doesn't impact all of it as much? Thank you.
Yeah, so how has the Nobel Prize impacted me? I'd say it has more of an impact on me personally than it does in terms of what we're doing in the lab. Because as I say, that what we're doing in the lab is largely governed by the interests of the people there. I've been committed to a small lab, so, you know, although people have contacted me in recent months from all over the world saying, can I join your lab? You know, I'm not taking in any more people, so it hasn't really changed that, you know. And also funding is limited anyway, and as I'm sure you know, the funding is in crisis mode in the United States right now. So many labs, not mine not yet, but are contracting, and because of people being let go. So I think one of the differences in my life is, like what's happening right here in this moment, is because people seem to be interested in what I have to say. And that has been something that, you know, I've had to adjust to. But I think for the most part I've welcomed, because I find that it's an opportunity for me to learn how to speak about a whole variety of different things that people are interested in. It's forced me to think about, actually, you know, policy matters. And at my university, because I'm the most recent Nobel Prize winner, I'm called upon, you know, to meet with the chancellor and meet with the dean, you know, about different matters, especially during these crises that have been happening. And I'm happy to do that. But it's not like I'm an expert, but I'm the Nobel Prize winner, right? And so I might be able to then take, you know, an idea to another level, you know, to the state and things like that. So the other Nobel Prize winner at UMass is Craig Mello, co-discoverer of RNAi. And he's the person who recruited me to UMass. He was my student at Harvard. So this man was my student. He won the Nobel Prize, and they recruited me to UMass. And so the two of us are kind of have sort of unofficial, you know, roles as trying to mediate, moderate, help think about issues on campus. It's a very interesting situation. But I welcome it because, you know, what you want to do in your life is somehow have an impact, right? And as you get older, you start to think, man, my impact is kind of evaporating, you know, and eventually it's going to disappear, right? So it's if somebody asks me to step up and try to help out in whatever context, and it's something new, I see it, you know, now as a learning opportunity, and I'm happy to try.
Hello, Professor. Congratulations once again.
Thank you.
I rather have a very brutal question, probably bitter, sorry for that. But as a student, I'm really concerned, and from my day-to-day basis, I have this question related to the integrity and value and trust in the recent scientific publications, especially in the era of generative AI and the growing publications. So that's my question, like how do you evaluate and maintain trust with this?
Well, that's a very interesting question. I mean, the broader implications of your question pertain to all sorts of contexts. How do we find and identify reliable sources of information? This is a problem in the public as well as the scientific career, sphere. Particularly about AI, I'd be very curious about all your thoughts and other people's thoughts about this, because, I mean, in one dimension that I'm wondering, maybe perhaps you're most concerned has to do with scientific publications. That's right? Yeah. And so, for example, generative AI can write a paper, right? And people have done these experiments, and you just give data, and it can write a paper. And so one wonders then, how, if a paper you're reading, was this generated by AI, or how much of it was generated by AI? Now, there's authors listed on this paper, but how much of what I'm reading was actually generated by the minds of those people? Before, we could be pretty confident that it all came from somebody's mind. And my view of this is evolving, and I'm trying to learn from younger people. So, for example, people in my lab who are not English speaking, English is a second language, they will write a manuscript, and then it would come to me, and then my job is to fix the English, right? And it's not just a matter of fixing grammar, it's fixing exposition, right? Clarifying. I know what the student is trying to say here, right? But a reader would not know what she's trying to say, because there's so many possibilities encompassed in the way it's worded. So then I would work really hard on that. It could take 2 weeks sometimes for me to work on somebody's manuscript. And then I'd procrastinate, because it's so hard work. And so finally, we realized, well, there's AI tools online which you can just take your rough drafts and turn it into better English, right? And that saves that student an enormous amount of time. She doesn't have to wait for me to do that, right? It instantly comes back. And so that saves work for me. So I finally said to people in the lab, let's do this. Let's use Edit GTP, and it will produce a marked up version of your manuscript, and then you send that to me, and then I'll work on it, and that saves me a lot of trouble, and so on. So I'm starting to understand now how AI can be used in a way that's actually positive, right? Now, what I worry about is that will students not really learn to write well, right? Because they're not going through the process of being sort of forced to write well, right? And I'm less worried about it now, because I realize, well, if these tools are always around, then maybe this person never has to become, like a professional English editor in order to be a scientist, right? And so I think that's great. There's ways that people can generate more effective grant proposals, let's say more efficiently generate papers and so forth using AI. But here's where I think that we as leadership, especially as a university, let's say, I think we're obliged to try to clarify for students exactly what our expectations are, or what the expectations are implied by publishing something, right? So if I publish something, you have to think of it as, this is my testimony here. This is my personal testimony. Now, much of it may have been generated by AI, right? But I'm responsible for everything. I'm personally responsible for how the reader interprets this, how the reader may misinterpret it. You know what I mean? How perhaps the way it's worded might suggest something that I don't mean to say, I don't want the reader to go that far, you know what I mean, in interpreting something, right? I want them to stop right here. So that clarity of intent, right, is something we have to take ownership of. So I don't know how we sort of teach students to do that, but I think maybe the principle is that everybody should be aware that somebody said every mark you make is you, right? And so everything you write is your testimony, you know? And so, you know, so that's, perhaps that helps people reflect on, all right, is it okay for me just to generate this thing and just plunk it down on the PI's desk and say, oh yeah, I wrote it, you know? I don't know, it's really, I think this is an evolving thing. And when I go to universities, you know, and meet with people, everybody's struggling with this. Even in high school and even in middle school, the teachers say, well, they're generating stuff by AI, we can't stop it, you know? So it feels like maybe this is something that we're going to have to work hard on for a while. Changing so fast, but we have to, yeah, we have to not ignore it, right? I think your question is really in that spirit of, here's something that we shouldn't ignore, right? Yeah. I'm sorry that my answers go so long, but that's because you guys, the questions are really good and it's fun to think about these things. Yeah.
Just continuing following this question about AI. There is a question, what is the aim of science? The aim of science is to find the laws which just govern the nature, somehow, us and other elements. And now there is a question, at which moment AI will be able to notice something new in the things which are produced by men, by experiments. Because at this moment, I think that your Nobel Prize, this is a lot of the thing that you noticed something what was against the established notions. That it was described in books, that gene, this is DNA, and this must be, and then proteins, and so on. And if you noticed something what was against, and you had this courage to say that loudly, yes, and to show this was something extraordinary. And the question is if AI will be able to do things like that, to notice something new which is against this what is written. At the moment, probably not, because there is knowledge which is established by human brains. But at some point, that's a question.
Yeah, so that's really interesting. A very provocative question, because what is the aim of science? And so you can imagine, let's say, a point where there may be a whole organ of science which is entirely AI, and the publications are basically AI shouting out sort of interesting insights that the AI has made, right? You know, and related to that, though, is this idea of looking for connections. Let's say looking for noticing something and calling attention to it. And I feel it's really, really exciting, right? And maybe you guys are aware of these sorts of tools, and I think there's at the National Cancer Institute, there's some people developing this. Basically, the idea is, what if I could read all the literature every day, right? And now, especially stuff that would catch my attention, you know, I can't even read a tiny fraction of the literature that's out there that would catch my attention, right? Because that's something I might be curious about or might vaguely relate to what I'm doing. So imagine you have an assistant that does that. Every day it reads all the literature, and it's learning as it goes what catches your attention. What are the kinds of things that you'd be intrigued by, right? What are the kinds of potential connections, right, and inferences and possibilities that you would like to have somebody call to your attention, right? And if it would learn that, it would be like, for me, it'd be like Super Victor, right? You can read all the literature and spot all the things that are worth maybe taking a second look at, you know? And even it might even sort of, you know, it would sort of make suggestions, the kinds of things that I might come up with over the course of a week, right? I'm in the shower, and I come up with some, oh yeah, maybe whatever. Or I'm going running on a Tuesday, and I'm running and I get an idea. And I said, that's right, it might, blah, 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 that might be cool. I should go back and compare those two things. And then maybe 2 weeks later, I happen to be driving and I come up with another idea about it, right? So what you'd like the AI to do is just do that in 1 second. Come up with those things that you might have thought of, right? But it would take you time. So it's like being super you, right? So that's really, I think that would be tremendous if all of us were like, had this agent that enabled us to be the super us, Yeah. Why not? This would be awesome, right? And then you can share these, and that's what they're trying to do, the NCI people thing, is you can actually share these things. So you might have connected to somebody else's agent, you can have it feed you stuff too if you find that you and that other colleague have similar interests or maybe complementary interests. Like you could say, let me have this interdisciplinary thing where this person who's I don't know, some theoretician of some kind, and then you're doing something else, and I'd like to have those connections come across my desk every day. Yeah. I mean, these are projects that could be a research project rooted at universities, couldn't they? And I think one of the things we're talking about is having each university has its own on-site large language model, or now there's these new small language model, where the data that's fed into it has been curated by humans in the sense that you guys would identify, let's say, all the journals you believe in, right, that are good, and good integrity to their editorial, and then you might even collect 1,000 papers that each one of you would contribute 5 papers and say, these are really well written papers. I mean, really literate, really super well written. And then we train our models on this, right? Not on all the junk that's out on the internet. So I understand there's great potential for that, so that each university, and that could be insulated now behind a firewall, so you're not revealing potential IP when you put it out there. Right now at UMass, you're not allowed to use ChatGPT actually, but people do it anyway. And because when you put out a manuscript that you're writing, now that potential IP is out on the internet, And they've identified cases where it circled back, and the development office spotted something out there, and it was because somebody had it rewritten by an agent. So having it insulated in your campus is probably important down the road.
So maybe that's why we are building here our local networks, scientific networks, something like this, to share our small parts of knowledge, and then we try to build something better, more, and maybe modeling will help us with this. Maybe it will be some kind of AI, but here, local.
Yeah, so you would, I think for example, I think you can get this service from Microsoft, but it costs a fortune, right? So not surprisingly, they can say, we can set up a campus-wide, insulated, AI system, and you can train it on material that you selected, so it's high integrity, related to your field, and all that stuff. But it's like super, super expensive. But anything in the, in the computational field, it can be done actually by high school students probably. So if you hire some high school students, they can probably set it up for you. I'm exaggerating.
All right, Professor Ambros has a lot of other obligations, so this is the last opportunity to ask a question, 1 or 2.
I'm happy to stay as long as you want. Yeah. There we go.
Hello, Professor. What is your assessment of H index and the citation and the list of Stanford of most cited scientists? It is a true indication of scientific research or not?
Could you please repeat the question?
About H index, evaluation of papers.
Evaluation of papers?
Yes.
Yeah.
It is a true indication of good research, scientific research or not?
Ah, yes, right.
Or second question, what is your H index?
Is my what?
What is your H index?
H index.
I do not know my H index. Yeah, I don't know my H index. No, I'm sorry. Yeah, so this general question is an interesting one. I think it's evolved, but maybe not so much. It depends on where you are and what your contexts are, right? So, so there was progress when bioRxiv came online, you know, because bioRxiv finally allowed, you know, the biological field to have a place for placing and publicizing manuscripts. And physics has been doing this for decades and decades and decades, right? And so finally, you could get credit for a discovery, and that includes priority with regard to patents and so forth, by publishing a manuscript that's not reviewed yet, right? I think that was a super, super helpful breakthrough. And the NIH and NSF in the US officially recognizes a bioRxiv publication as a real publication, you know? So that's really, and so you can include in your productivity, you know, over some period that you're being reviewed, those manuscripts that are not yet in press. They're in review, but there they are in bioRxiv. And so I think we're moving. There's you know, releasing perhaps some relaxing of this sense, the traditional, let's say, you know, a point of view, which is that the papers that are valued are the ones that are in this, you know, top tier journals, right? And really, the important thing is the journal that you published in a journal with strong editorial integrity, right? So therefore, somebody can say, oh, that's, you know, for example, genetics, you know, the journal Genetics. It's a good journal, really good integrity. But now, in the same time, there was a period of time which thankfully is no longer is fading. There's a time when the journals sell. That most of the papers in there you essentially could not believe, really. And so that's because they were publishing stuff that looked flashy, and then stuff was written in a way that was flashy, stuff that, you know, the implications were there and even in the title, and then when you read it, wait a minute, the evidence isn't here. You know, this is a story, it's not a paper. So it's gotten a lot better now. I'm really excited by Cell now. So, but there's a lot of other journals that have always been solid, you know, the whole time. So I think it's very important for institutions to recognize that, you know. So my advice to young faculty, for example, is to publish often. Publish smaller papers more often than trying to do a big paper, you know. Because in the end, the arc, say, of your career is going to be the same. But having multiple publications like this rather than big publications here, you know, and here, right? It's much better. Yeah, and some H index. I don't know my H index. It's embarrassingly low, I'm sure. Yeah, actually.
Hi, thanks a lot. First of all, I want to say it's a great pleasure to be a part of this meeting and listen to your answers. My question is a follow-up to one of the previous questions about the things that we still don't know. So what do you think would be the next breakthrough in molecular biology? Or if this is too much of a fortune teller question, so which fields of molecular biology do you think it is worth to put effort into so that we can make a society a breakthrough? So
what you've done with your question is you will illuminate for everybody why I'm not a leader of an institution like this, right? Because I'm very weak in this kind of vision. I mean, I know what excites me from moment to moment, but I see that can change, actually, or not moment to moment, but from month to month. So that makes me doubt my own abilities to have sort of long-term vision because I'm excited by certain things at a certain time. So I told you, for example, we have a project in the lab where we're doing something new. We're working on a newer animal, this ACEL that does whole body regeneration. And so the postdoc and I and our other collaborator are curious about what is the contribution of microRNAs and other, RNA-based regulation to this regenerative phenomenon? Because we're excited that this is a process that is ancient, right? The common ancestor of all the bilaterians could do whole body regeneration. Why do we know that? Because planaria, way down here, can do it. The acial, way over there, can do it. And then sponges and starfish, and so it's a property that is sort of endemic across all the animals. And so then people say, well, humans can't do whole body regeneration, so why study the phenomenon? And that is so wrong, because we can regenerate tissues in an amazingly well. We do it all the time. We regenerate our skin, our intestine, our liver, and then we heal wounds. And so it's emerging that the mechanisms of regeneration in flatworms and acials are the same molecular fundamentals: Wnt signaling, this EGR, pioneer transcription factor. So as we learn about whole body regeneration, we're learning about fundamental mechanisms of tissue replacement that we do all the time. So that excites me, because I feel like, I really love everyday processes that are mysterious. And so when I talk to, I should have brought my slides, I don't know, but when I talk to high school students, I show them this, where my thumb, I do woodworking, and the router took a big chunk out of my thumb once, right? And so there's a big hole in the skin, right? And so I took a picture of it, and then I let it heal, and I have a time course of this thing, this hole in my thumb filling in with skin, and the fingerprint got restored, So I say, this is something that I can't explain to you how this happens. I cannot tell you how this, and we can tell you the rudiments of it. We know some of the gene products and signaling processes that are going on, right? But I can't tell you how it was perfect, right? It didn't make too much skin, it didn't make too little skin, right? And it didn't make toe skin or shoulder skin, it made thumb skin, And so I feel like it's a wonderful illustration of everyday phenomena that we take for granted, but are actually deeply mysterious, right? And I think it's cool that maybe we can learn fundamentals of that by studying this animal that can do whole body regeneration, And at the same time, maybe we'll learn something about evolution, right? Because we'll be able to compare the process in planaria versus, the acial and wound healing in people too. So was that an answer to your question? Yeah, right. So it's a matter of taste, isn't it? And so that's why you need to have a lot of people sort of doing what they want to do, right? Yeah, because you never know where you want to go.
Yeah. What are habits of a good scientist?
What are the habits of a good scientist? Yes, okay, here's the first thing we like to say nowadays is that everybody can be a scientist, and whatever your habits are, those were probably good habits, right? Now, that doesn't mean that we can't learn from each other, right? Oh, we can recognize, well, that person approaches this a different way, and actually, oh, I bet that would work for me, that kind of thing. So we can learn from others. So I think those are the two things that are going on, right, that are super, super important. One is each one of us believing in the way we think and the way our habits and the way we approach problems. That has great strength and validity, right? Why? Because it complements the way other people approach problems, right? Now but to make that work, we have to work in a social environment, right? We work on teams, right? We work as collaborators, and we learn from each other. So if you go to somebody's talk, right, and they're talking about their project, often I see, oh, that's a really interesting way of approaching a problem or asking a question, or, oh, that's a really interesting way to analyze data. They're doing it better than I would have, You see, so you can improve your habits by learning from other people. Yeah. So maybe the particulars of how you go about doing science are not as important, but the principle would be to try to always improve yourself, but improve the way you are, the way you think, and value that. So it's not important for me to try to think like you or you to try to think like me. In fact, we don't want to do that. But we want to talk, right? And collaborate and brainstorm and see what happens when we talk. Because I'm sure you've experienced this, right? You sit down with somebody and talking about it, and often the two of you came up with some idea that neither one of would have come up by themselves just thinking about it. So, yeah, the best, if you were to come down to one habit, I would say, for good science, would be to talk to other people. Yeah, share ideas and talk.
Yeah, perfect.
Okay, let's forget for a moment that you are the Nobel Prize winner. Okay. Probably you didn't have such moments since last December.
Yeah, okay.
And I would like to know something more about you. So, 3 questions. First question is that your father was born in a small village which have been placed over this year in Poland. So, did you have any stories came from your father about the Poland, about the history, about our tradition? So it is the first question.
Right.
The next 2 is, what do you like to do during your free time? And what do you like to, how do you like to spend your free time? Okay.
Well, I can start with the second one. I mean, I like to, I have grandchildren, so those are the moments with them are probably the most treasured moments that I experience now. I like to, just go out and run, and have some solitude, right? So, and I'm always trying to become a better guitar player, although I'm a terrible guitar player, so I try to get better. And I just love guitars, and I try to, as a hobby, I make guitars or repair guitars. So that's steel string guitars. So that's a valued habit. I also, love astronomy, so I read astronomy magazines, and read about the latest. I'm sort of, so that's sort of free time stuff. Now, so what your other question was about? My dad. Yes, my dad was born in a small village that's now in Belarus. It was Eastern Poland. He and his sister were orphaned when they were young, and so they had a pretty hard childhood. So my dad didn't really talk about his childhood, and now, in retrospect, I understand why, because it was difficult. He did share some things, but they were bouncing around from one uncle's and aunt's house to another, and things like that, And then his schooling was. I'm going to talk about it a little bit tomorrow, also. But his school was interrupted by the war. I mean, terminated by the war in high school. So he was a really smart person whose opportunities were cut short. So we were very conscious of that when we were youngsters, that here was a man who did not have the opportunities that we have, that he's actually, bringing to us. So it wasn't until his sister emigrated to the States in 1960 that we had a little Polish community, because she had 4 kids, and so we would get together with them, and then she had some other relations and other friends that came from the same region, actually of Western Poland, because they had been relocated, So then we had a community, and actually I learned more about Polish culture from my aunt than my dad. And primarily it would be, often we would go to her house, and we could even come there by surprise, and she would somehow have all these cakes, and dishes that she had. She could just pull them out and set them up around us on the table, and she would tell stories, of her childhood and her time in Warsaw during the war. And so it was, I did not have really a Polish cultural experience, certainly not as a youngster.
So I wanted to ask something about regeneration, because the story about the thumb and the fingerprints really brought something to mind. Like, fingerprints are really interesting for a regenerative reason, because they actually don't really rely on genetics. They are made in the during the pregnancy in the amniotic fluid. So even identical twins have different fingerprints. So how do you suppose those fingerprints are then regenerated the exact same way?
Yeah. So I don't know. I mean, I think I'm assuming there must have been a dermal layer remaining where my injury was, because I don't understand how. I mean, it was a very deep wound, but I didn't analyze, the wound and confirm that I didn't. For example, I could not see muscle or anything like that. So there might have been a layer, a deep dermal layer there that had the memory somehow of the configuration of the fingerprints. I mean, maybe you know about this, but I think I'm kind of assuming that must have been the case, because it was hard for me to imagine that if there was no information, how the surrounding tissue would somehow, maybe it interpolates those lines or something. Yeah, I don't know.
Do you think it would be an interesting area of study?
Yeah, I can't imagine getting approval for the experiments, though, right? That's a very good question, and I don't know if is there an analog to fingerprints on the in a mouse. So obviously, experimental systems like that, maybe opportunistic. Maybe there's information known about this from studies of injuries, studies of burns, and so forth. Yeah, what are the requirements for full regeneration of skin?
Thank you.
Well, I think one last question, because I have already got this serious look from the rector, trying to tell me that it's time to finish.
Okay, now I got stressed, but thank you. So very quick question, as young postdoc and to the experienced researcher, do you have any piece of advice how to be a good mentor and how to balance teaching and research in a clever and nice way?
No, I'm not good at that. Balancing teaching and research was always a challenge for me. I used to be, in an arts and sciences department where I had a significant teaching load, and I always found that the research was much more satisfying than the teaching, because I never really felt that I was very good at teaching. It always seemed like you could always put more effort into it, and you still never felt that you were adequate, right, with teaching. So for me, I think that's a reflection of, a lack of real talent for it, So, and how to be a good mentor, what I would say is that, when I started out in my lab, I was not a good mentor because I was not deliberate about it. I was not thinking about, how should I mentor this person, right? It feels like there wasn't in the culture a craft, a sense of a craft, mentoring craft, you know what I mean? And so at least I didn't sense it. And so I was always just sort of improvising, yeah. It's more of, I think maybe imitating people who seem to be successful. Maybe I think that's what I'm doing now, and I have been doing, and nowadays you can actually take mentoring. Yeah, you can go to mentoring seminars and so forth, and that would be something that I would have benefited from enormously in the beginning. So, all right. So, what's the bottom line? Avail yourself of whatever resources there are for mentoring, training, and identifying people around you who seem to be successful, and do what we were talking about. Just talk with them about it. It looks like you and your students are really, going great guns. How do you do it? And I have a colleague like that. She's only been there, 2.5 years, and she has zillions. I mean, it's all these students, and they're just doing so well, and they say that she is amazing, right? And so, when you hear something like that, maybe you can go and find out from them, But it could be that you're the person that other people should be talking to. So maybe you're already the expert in mentoring.
I think now you will be the most famous lecturer at the university.
All right, so let's thank Victor for his answers and talking to us.